Good evening, ladies and gentlemen. Welcome to the Melbourne Brain Centre and to this ICT for Life Sciences Forum presentation. My name is Luan Ismail. I'm the forum convener, and it's a delight to have you here this evening. As most of you know, you've been here, or you've been to some of these events in the past. The forum has been established by a number of research institutes, universities and companies in Melbourne to essentially promote this concept of the convergence between the physical sciences and the life sciences. And Melbourne's a hotspot for that activity, and really this is an opportunity for us to engage with the community, showcase some of the exciting research that's taking place in Melbourne and around the world, and how it's impacting or can impact us, will impact us in the future. This evening we've got Professor Phil Hodgkin from the Walter and Eliza Hall Institute of Medical Research. I'll introduce him and we'll get the event underway. So Phil is an experimental and theoretical immunologist and currently is the head of the immunology division at WEHI. His primary research interests centre upon the regulation of T and B lymphocytes. His laboratory develops quantitative methods for exploring this question, including single-cell imaging and division-based tracking. Phil studied microbiology at the University of Western Australia and obtained his PhD from the John Curtin School of Medical Research in Canberra in 1985. He then undertook postdoctoral studies at the DNAX Research Institute in Palo Alto, California, where he broadened his initial interest in cytokine production from T cells and the regulation of antibody production and isotope switching of B lymphocytes. In 1990, he returned to Australia to take up a research fellowship at the John Curtin School of Medical Research, and in 1995 he was appointed group leader at the Centenary Institute for Medical Research. In 2000 he moved again to the WEHI, and in 2006 was appointed head of the Immunology Division. Please welcome Phil Hodgkin to deliver his presentation, Reverse Engineering the Immune System.
Thank you very much, Luan, and thank you very much for the invitation to present at this forum. I'm a little daunted because the list of potential people that are here from different disciplines has made me wonder how I could cover and give you something interesting for all of you. So I've tried to do that. And the way I thought I would attempt to do that would give you a little bit of the history of the subject of immunology, particularly leading up to the founding theory, the great theory on which even our current understanding of immunology is still based. And I think you'll find that interesting if you haven't heard it before, because it has a local flavour, a local connection. And then, having got to that point, we'll run ahead and I'll show you some of the exciting things, hopefully, that we're doing in the discipline, in the area that Luan mentions, that if you like, the theme of this forum, which is to find common connections between other disciplines. So we've been working hard to combine immunology with computational biology and mathematical modelling in particular. So let's get started. So building up to this great theory, I'll take you back to 430 BC, to the time of the Peloponnesian War. And it was written about by Thucydides, the historian. And in the second year of that war, the plague hit Athens, and a quarter of the citizens of Athens died as a consequence of this plague. And Thucydides wrote in his history that the sick and the dying were tended by those who had recovered, for no one was ever attacked a second time, or not with a fatal result. And this is the first record we have of an observation of immunity. Okay. Now, we don't know what that plague was. It probably wasn't smallpox, but smallpox is one of the great plagues that devastated Europe for centuries. And this is a picture of a patient suffering from smallpox. You can see the terrible pox and scarring that would occur as a consequence, even if you survive the disease. And the other point to make about smallpox is the incredibly high mortality and infection rate. So 20 to 40% of people who had the major form of this virus would die. 80% mortality in children. And in the 1800s in Europe, it's estimated to be the leading cause of death. So this is a terrible scourge of mankind. And there's a great story here, and it emanates from this country physician in England, Edward Jenner, who just before 1800 did a key experiment which centred around an observation he made, which famously was because he noticed that the milkmaids, the dairy maids that attended the cows in the countryside, were renowned for their beauty. They didn't suffer, it seemed, from these pockmarked scars from survivors of smallpox. But they did often get infected with cowpox. And so Jenner put two and two together and thought, well, maybe the cowpox infection is protecting them from, which was a very mild disease, was protecting them from the very severe disease of smallpox. So he did an experiment. He took an infected cow called Blossom, who had infected a dairy maid, Sarah Neames. He took the pus from Sarah Neames, from that infection, and he introduced that to the volunteer. Well, I'm not sure he volunteered. This was a young child here, the son of a friend of Edwin Jenner, and he introduced that cowpox virus into his arm, and sometime later, a few weeks later, actually infected young James with smallpox from a infected patient. And fortunately for both James and Edward, he survived. And this became the basis for the first immunisation. So the strain that he developed from cow was called vaccinia, from the Latin, vaccae is Latin for cow. So this was the vaccine strain. And Jenner predicted as a consequence of this treatment that one day the annihilation of smallpox, the dreadful scourge of the human race, will be the final result of this procedure that he had developed here. And the amazing thing, the great thing here, is that this turned out to be true. So here are the two last natural cases of smallpox in the world. This is the major form, this is the minor form. This is a young child in Bangladesh, an adult in Somalia here in 1977. And in 1980, Frank Fenner, who was charged by the World Health Organization, an Australian virologist, to monitor the success of the campaign, declared to the Assembly that they declare solemnly that the world and its people have won freedom from smallpox, a devastating disease, sweeping an epidemic form through many centuries since earliest times. And this was a great triumph of this technique. The next part of the story, jumping ahead, Louis Pasteur developed the germ theory. So he realised that infectious disease, different infections were caused by different types of organisms. So polio would be a particular type of infection, tuberculosis would be a different type of germ, tetanus would be a different type of germ. Each disease would have a different type of organism here. And he put two and two together, that if the immunity is specific for the type of organism, he could use Jenner's method to induce immunity to each disease. So here's was his strategy. He called it vaccination in honour of Jenner. Identify the disease-forming organism for the infectious disease. Develop some weakened strain, capable of inducing immunity but not capable of causing the disease. And use that to stimulate against the potent form. And as a consequence of this strategy, Pasteur developed a few vaccines himself, but very quickly, in the ensuing decades, as a consequence of Pasteur's work and germ theory here, rapidly many infectious diseases became protected by the development of these weakened forms, these vaccines. And I'm not going to go through these in detail or tell you about the great success. They've all worked extremely well. And of course, recently we have an Australian success story here, the human papillomavirus vaccine developed by Ian Fraser here in Australia and is now widely used in protecting against cervical cancer. But this success of vaccination is based on the germ theory of disease, the experience of immunity, but it doesn't tell us anything about how the immunity was working. And you didn't need to know that to implement this policy and develop these vaccines, interestingly enough. So how does immunity work? And this is the point of reverse engineering. Now, my good partner Lindy over here, my saw the title of my talk, he said, I don't know what you're talking about. What's reverse engineering? So I thought I would just remind you that reverse engineering is the process of discovering the technological principles of a device, object, or system by analysing its structure, function, and operation. This is straight off Wiki. It often involves taking something apart and analysing its workings in detail, or trying to make a new device or program that does the same thing. And why I like this idea is that this is kind of the scientific problem. This is one strategy, experimental science, pulling it apart. And this is, if you like, the ultimate goal, developing a theory or a model that we can use to simulate or recreate the complex system in a different way. So this is reverse engineering the immune system. It's really the scientific investigation or the science of immunology that I'm going to talk about. And so here are the steps that lead to the theory underpinning modern immunology. So we've seen immunity working. We see how powerful it is to protect you against infectious disease. But what's happening? Well, the first key bit of information was provided by a German scientist, Emil von Behring here. He was a student of Robert Koch, a great bacteriologist in Germany. And he discovered that having infected a guinea pig with diphtheria toxin, that something appeared in the serum of that guinea pig, that you could transfer the serum from the guinea pig to another guinea pig who had never seen diphtheria and they would be protected. Not only that, you could take the serum and add diphtheria to it in a test tube and the diphtheria could be killed in the test tube. So something appeared, as a consequence of being exposed to the diphtheria, something appeared in the blood, in the serum, that was capable of attacking the bacteria. So he reasoned that now, as a consequence of exposure, serum now contained antitoxins, which we call antibodies. Now, so this was in the late 1800s. We didn't have a comprehensive theory of how antibody was working until the 30s. So what happened in the 30s? It was discovered that antibody was a protein, it was a gamma globulin. You could recognise it in the serum. It was realised that the way they were working was that they were complementary in shape to the toxin. For example, this is illustrated here by the classic lock and key mechanism. If you can imagine, this is your so-called foreign material, the antigen. It would have a certain shape at its surface, and the antibody would make a complement to that shape, allowing it to fit together and bind it and neutralise it in that way. But the interesting point here is that Landsteiner, an Austrian immunochemist working in the US, made shapes that had never existed on Earth before. He was a chemist. He used his chemistry knowledge to make shapes that he knew had never existed anywhere before, and he could still make antibodies of complementary shape. So it seemed apparent that antibodies apparently have some unlimited specificity against any shape. And because of this last point here, it was very challenging to think, how is it that this protein could have this magical property? And the earliest theory was proposed by Linus Pauling, who won the Nobel Prize twice, once for chemistry, once for peace. But Pauling championed an idea that the antibody was a special protein that could wrap itself around the foreign material. The foreign material would act like a template. So it had the magical property, wrap itself around like a mould. The antibody would come off, it would have an imprint of the antigen and would go off into the serum, and then some process would wrap a new molecule around and send that off into serum, wrap a new one around, send that off. And the idea was that, so by changing the template, you could wrap it around and get a different shape in this way. So that was the theory. This was the dominant idea, very dominant for 2 decades, about how immunity was working, how antibody was working. But it was wrong. And eventually it was replaced by the correct theory, the one I'm going to tell you about, which is a completely different type of theory. You had to think it completely outside the box to come up with this idea. And the person that did it here in Melbourne. This is Macfarlane Burnett. He was a virologist, he's working here with viruses, but he became the director, as you'll see here. He became, in 1945, the director of the Walter and Eliza Hall Institute across the road. But he grew up in country Victoria in Traralgon. He did a medical degree in Melbourne in 1924, did some postdoctoral work, PhD studies in London, in the late 1920s, and he made a seminal decision, in my view, one of the most important decisions made for Australian medical research, is that he decided to refuse offers from Harvard and Oxford that he had to work. And most Australian scientists of those times would work overseas. Florey, for example, worked overseas all his life. But Burnett decided to come back to Melbourne, to a position at the Walter and Eliza Hall Institute, where he established a great reputation as a virologist and geneticist. And he was always interested in the problem of immunity, the specificity of antibody. And in 1949, he wrote a book with Frank Fenner, the same Frank Fenner that announced the eradication of smallpox. Fenner was a protege of Burnett's. And in that book, he challenged the Pauling's template theory. He didn't know what to replace it with, but he just pointed out some logical problems with it. One was, how could it tell the difference between complementary shapes that were in your own body versus what was a bacteria? There was no mechanism by which you could tell the difference. In fact, the whole concept of self, non-self, which is very central to our understanding of immunology now, was invented by Burnett in this book. And in fact, he got the Nobel Prize for a single sentence in this book, suggesting that maybe you learned self-tolerance as a newborn or very early in life. And as a consequence of that, Peter Medawar in London did some experiments for which he demonstrated that point, and they both earned the Nobel Prize in 1960. So self from non-self. And the other point that Burnett pointed out was this graph, where he measured antibody production over time after being exposed to a virus. Now this is a logarithmic scale here, so this is an exponential increase in antibody. And he said if you're making antibody on a template, you expect a linear increase in production over time. And you're not seeing that, you're seeing something exponential, which to him as a biologist, he realised must involve some growth, like he would see in his bacteriophage cultures or his virus cultures. Okay. So he pondered this for quite a few years. And in 1957, he wrote this paper. And this is the paper that contains the theory. It's called clonal selection theory. It's, in my view, the most important and the greatest scientific theory in biology of the last century. And we can argue that. But there's a couple of reasons why I think that's true. But just to give you some of the background, it's published in this incredibly obscure newsletter of the Australian and New Zealand Association for Advancement of Science, of which he was president of the time. So he had inside running to get this published. The reason he published it here is because he wanted to establish priority. He thought it was such a crazy idea. If it's wrong, no one will read it. If it's right, I've published it. But in this 2 pages, and it's really, this is all introduction, it's in this second page here, all the principles of this important theory are laid out here. And it's interesting to look at the paper that follows it, some processes of the Pacific Island peoples in handling and storing foodstuffs. So here's the greatest biology theory of the 20th century, followed by something not quite so important, perhaps. Okay, I'm going to firstly tell you that very quickly Burnett realised that this theory was right. And just a year later, his student who just arrived, Gus Nossel, many of you will know him, we have the Nossel Institute now across the road here, did a key experiment demonstrating an important point of his theory, which we'll talk about in a second. And within 10 years, it was clear that this theory that he put forward, he called it the clonal selection theory, was right. Now, there's a lot of consequences of that. One was, early on, he brought in all the staff of the Walter and Eliza Hall Institute and told them that as of today, we're not going to be a virology institute anymore, we're going to do immunology. Because he realised that he had a head start against the rest of the world to pursue this really important subject. And that proved to be critical for the establishment of the WEHI in Melbourne and Australia to make a real impact in this important area. Okay, now what I'm going to show you. So that was 1957, he published that paper. So in 2007, 50th year anniversary, Walter and Eliza Hall Institute, we have a great graphics illustrator there, Drew Berry, and he helped us put together a movie illustrating the concept. So for those of you who haven't seen this, I'm going to play it now because I think it does a much better job of explaining the principle than I can in words or pictures. So let's just take a second to watch this movie.
In 1960, Sir Frank Macfarlane Burnet was awarded the Nobel Prize for his contribution to the field of immunology. Burnet's clonal selection theory revolutionized our understanding of the immune system. To this day, Burnet's research remains the foundation of the way we think about disease and how the body responds to it. Clonal selection is the central mechanism by which your body fights infection. The following visualization presents clonal selection during an infection by strep throat bacteria. This patient has a strep bacterial infection causing a red, sore, pussy throat. The tiny round strep bacteria thrive on throat tissue. The bacteria are coated with hairs called M protein. M protein is what your body recognizes to be foreign. In response, your body will launch an immune attack against it. Your lymphatic network is an important part of the immune system, detecting any infections that arise in your body. The M protein hairs drain into the lymphatic system around this patient's neck. The lymph node is crammed full of B cells. B cells are guardians of your immune system, scanning the body for foreign material. Arriving at the lymph node are M protein hairs from the bacteria. Your body contains billions of B cells. Each B cell is unique and is sensitive to only one kind of foreign infection. By chance, one B cell is sensitive to the M protein hairs from the bacteria. This is the molecular surface of that B cell, magnified 10 million times. Its surface is covered with receptors that recognize M protein. The receptors grab onto the M protein hairs. This triggers an activation signal to travel into the cell. The activated B cell migrates and begins to replicate. In a few days, thousands of copies of the selected cell are made. This is the central idea in Burnet's clonal selection theory, where one cell is selected out of billions of B cells and identical clones have been created from it. Therefore, the cell is clonally selected. This creates an army of B cells that mass produce M protein antibodies and release them into the surrounding fluid. The antibodies circulate in the blood and travel into tissues. They bind to the M protein hairs on the strep bacteria. This flags them for destruction by the immune system. Other immune cells ingest the antibody-covered bacteria. They destroy the bacteria and eliminate the infection.
So Drew Berry and Etsuko Uno prepared that for WeHi TV. If you're interested in this for educational purposes, you can download it off the web or YouTube. Okay. So let's just quickly run through some of the details of that, because some of it was left out. If we think of this as the antibody molecule, the protein, it's got two arms, antigen binding sites here. And the idea is that the surface here is the surface that interacts with the foreign material and it makes a three-dimensional shape. And that antibody has two lives, one as a soluble molecule like this, where it can circulate in serum, and the other as a receptor on this cell here, like this. And so by selecting this cell, out of the billions, this cell can be stimulated to start dividing. And it's because of that division that the production of antibody makes that exponential increase that Burnett alerted to Burnett that a new theory was needed. So, but the other aspect of the theory that wasn't discussed so much in the movie is a key point, which is how is it that this protein molecule could be designed so that it can recognise millions of potential, this very broad range of potential possible surfaces? And this is the key. This is what makes this theory so great, and it's a single sentence in the paper. Burnett's idea was that, and this is what he wrote in the paper, we must hypothesize a genetic process for which there is no available precedent. This is 1957, not long after the DNA structure has been identified. A randomization of the coding responsible for part of the specification of gamma globulins, antibodies. Randomization. He realized that the power of randomization could do the trick. He had no evidence whatsoever for making this prediction. So this turned out to be absolutely correct. And this is Susumu Tonegawa, who showed in the 70s that indeed the final antibody molecule was made up of bits and pieces chosen from a range of possibilities. So for example, one of the parts of the protein, the heavy chain, has to choose one of these, one of 65 here, one of 27 here, one of 6 here. So we get 10,000 possible variants of those. That has to combine with either one of these or one of these, and there's 2 possible chains at which they can be formed from. So the total possible specificity, just in all the combinatorial possibilities here, is about 3.5 million. There's other enzymes that nibble and tweak the process, increasing the possible number of specificities, well over probably 100 million possible surfaces. So the amazing point here is that just by 178 bits and pieces of genes here, you can code for over 60 million different specificities. So a massive efficiency in the generation of the possible surface, the possible different types of antibody. But of course it means that you can't simply have an immune system working from a single cell. The only way it works is to generate the population, using that randomizing process to generate individual cells, each with a unique specificity that they are guardians of. So by making a large number of otherwise identical cells, apart from the receptor, your immune defense has the capacity to recognize anything that it's exposed to. Now, I'm going to leave the story of antibody soon. I just point out that the key players in the antibody story, and we haven't talked about all of them, but von Behring, Paul Ehrlich, his colleague, Landsteiner, who made those artificial antigens, Yerner, we haven't talked about, but he provided a key bit of information to Burnett, Tonegawa, who showed the recombination of the receptor. And Milstein and Curler we haven't also talked about, but they showed that you could take the whole concept of clonal selection and immortalize a particular clone and make a pure version of an antibody, a monoclonal antibody. And this was a great development. And even today, it's incredibly important because this is now a billion-dollar drug industry. Antibodies are very well tolerated as drugs because they can target not just infectious agents, but cell surface like tumours or usually rogue tumour cells or transformed cells in some way. So this has become very important too. All of these individuals have been awarded Nobel Prizes for medicine and physiology. So it's an incredibly important story, not just intellectually, but also for contributions to health. Okay. So let's run quickly ahead. We've looked at this beautiful theory, but 50 years later, I can tell you, as we teased it apart, it turned out to be much more complex than Burnett may have imagined. Where does the complexity come from? Well, there wasn't just B cells, there's multiple types of cells that can respond and start proliferating. We call them T cells, in general T and B cells. The T cell was discovered by another Australian, it turns out, in working in London doing a PhD, Jacques Miller, discovered the role of the thymus. Take out the thymus, you get no T cells and you suffer from many infections. How T cells, what they recognised was identified by Peter Doherty and Rolf Zinkernagel working in Canberra. Peter Doherty you will know, he's also Doherty and Zinkernagel won the Nobel Prize for this discovery, and Peter Doherty has been Australian of the Year recently. And there's an institute being built just across the road named the Peter Doherty Institute as a consequence. So T cells turned out to be very important in addition to B cells. Think of the difference. T cells tend to monitor what's going on inside cells, whereas B cells are monitoring what's happening in the fluid spaces. So you have these two major arms of the immune system looking inside cells and looking at the fluids as a major categorization. The other complexity here is that cells turn out to communicate a lot. They're autonomous, but they talk to each other. T cells talk to T cells and affect their behavior. T cells talk to B cells and affect their behavior. T cells talk to, they need cells to present foreign material to them, to tell them, to get them excited. And one of those cells is called a dendritic cell. Discoverer of this cell, Ralph Steinman, was awarded the Nobel Prize last year for this discovery. So that's part of the complexity in trying to reverse engineer what's happening. The fates that cells can take on are highly variable, and I'll show you an example of this in a second. They don't just proliferate and attack foreign material, they have to make decisions all the way along do I respond or not? Is this self or not? If I respond, what am I going to do? And then, as you saw from those lymph nodes and the migration of the cells through the body, there's complex positioning of cells that make an important impact on the effectiveness of the immune response. So this is a little summary of what makes immunology complex. Nevertheless, of course, there are huge health benefits for understanding the immune system that we could intervene. Obviously, development of vaccines and tackling infectious disease, even the hard ones like HIV or malaria, they're hard. They're proving hard to make vaccines, but we're making progress. But it turns out that many diseases arise internally. So if you make bad decisions about what's self, you can attack yourself. Your immune system is responsible for so-called autoimmune diseases, and the best known examples are perhaps type 1 diabetes, where you attack your own pancreas, or multiple sclerosis, or rheumatoid arthritis, or lupus erythematosus, all examples of autoimmune diseases. Allergy and asthma have an immune component where you make an overreaction to something innocuous, so pollen or peanuts. This is an immune response as well. Tissue graft rejection prevention. Tissue graft rejection is also a powerful immune response, which if we could control, obviously would have major health impact. And increasingly it's becoming apparent that the immune response is playing a role in protecting you against tumours. Tumours that are arising spontaneously are often being dampened by strong immune processes. So major health benefits to be gained by being able to manipulate positively or negatively your immune response. Okay, so here's our challenge. As immunologists in the modern day, our challenge is rather large. To understand the biology of a complete complex system. The immune system is very complex, and it's complex because it operates at multiple levels of scale. So I've tried to draw them here from the molecular, what's going on inside the cell, the genome level, to the signaling level, to the way cells communicate with each other in these bundles by moving past each other, how they collect in different organs and transmit themselves around the body, and then ultimately providing protection for the whole organism. So we have to understand this very complex system at all these levels, and at every level it's complex. Almost imagine the closer you get to the inside the cell, the more complex it becomes. And immunologists use many different cellular and molecular methods to dissect this complex system. And I think it was fair to say that they're at the forefront of these types of techniques. They can image cells, they can track them, they can trace them, they can remove genes and study the consequences in very clever ways. Now, and I should say, still 50 years later, I'm proud to say that Australian immunology, particularly in this campus, is holding its own in this kind of technology and dissecting this important health system. Okay. But do we know enough now to follow the final result of our reverse engineering goal, to try and make a new device or program, a simulator of the immune response, a theory that captures everything. Now, what I want to do now is show you some insights I think we are deriving across the road that gives us hope that this is going to turn out to be simpler than it at first appears, and that we're making good progress, and very optimistic about the opportunity for using computational tools to solve these problems. Okay, so what I've shown here is just the simple, I like to call it canonical. It's what happens when the immune response gets started. So you start with lymphocytes, they're naive, they get activated. As we saw in that movie, they get bigger, they start dividing. As they're dividing, they differentiate, they change the type of cell, they develop effector functions. And while they're doing that, there's lots of decisions being made. Eventually, so this is our cell number there, cell numbers increase. Eventually they stop dividing, having made lots of different types of cell. Many of them die away, having eliminated the infection. And some of them die forever. Some of them stay behind and live for a long time, either as memory cells to protect you against reinfection, or they may become long-lived plasma cells that go into the bone marrow and just make antibody. It might be antibody against flu for the rest of your life that provide protection against next time that you see flu. So this typical response, massive proliferation that takes place over 3 days to a week, cessation and then death, leaving behind a population that are going to give you protection in the long term. Okay. But to illustrate some of the complexity, here I'm going to use B cell fate, and I'm going to talk about the types of antibodies. So here, the colour in this picture here was supposed, in the movie, is illustrating the different antigen specificities. The colour over here is a different feature of antibody. So we can take the same B cell here, and it will always make one particular type of antibody at a time, but it's capable of switching between them as a consequence of some information. So they always start out with this type of antibody. They can switch to IgG, 2A, 2B, 3. This is mouse antibodies, but it's the same story for human. And having chosen a particular type of antibody, it can change its cell type. So it could be dividing what's called a lymphoblast here of any of the type of, making any of these type of antibodies. It could become a memory cell of any of these types. It could become a short-lived antibody secreting cell or a long-lived antibody secreting cell. In reality, the heterogeneity, the number of different fates that B cells can become is even greater than this, because there's subsets of these. But I'm just trying to illustrate part of the issue is that in any given response, the cells get activated and then they start choosing different fates. They don't fill out all those possibilities, there's just a small subset of them. So how is it that those cells make decisions about their fate? And I'm going to tell you two main theories. The first theory, which is, if you like, the one we've been operating under for a long time, is that cells receive cues to direct their fate. So here's two identical cells. This cell gets a signal, a hormone that tells it to become a different cell type. This gets a different hormone and it becomes another different cell type. So this might be IgG1, this might be IgA, for example. Here's a cell that's dividing. Maybe it's getting a growth factor to tell it to divide. This cell goes on to die. Maybe it died because it failed to receive the growth factor, or maybe it interacted with some other signal that told it to die. So you can think of cell fate and directing cell behaviour as coming from outside the cell, as being told what to do at every level. That's a popular version of how cells are controlled. And there's lots of evidence that, in fact, you can control lymphocytes. Even in vitro, we can generate lots of interesting behaviour. So here's our IgM positive cell, and by culturing them in different ways with different, these are cytokines produced by different cells in the immune system. These can direct cells into these different antibody types. So that's consistent with that hypothesis that you have to tell cells what to do. The problem with that idea is that to solve this complex system, if you think about it, we have to have a very sophisticated understanding of where signals are coming from, where cells are getting direction about whether to proliferate, whether to differentiate, whether to die, and where are they getting those signals, and what's coordinating all of that to make it all work effectively and appropriately every time. It's a tough problem. Okay. But there's an alternative that I wanted to argue for here. And the alternative is more like a wind-up toy, shown here by this bus. The automaton, like a little robot. You wind it up and away it goes and does a few things and then runs out of steam. And I think, so there's some evidence that this is what's actually happening. So this is how it worked for cells. We might take the cell and expose it to a set of signals here as an undifferentiated cell. And as a consequence of that activation and exposure to those signals, the cell is effectively programmed to divide 5 times, stop, and then die. So it would do something like this. Divide 5 times, stop, and then die. And as a consequence of that, we'd get growth, cessation, and death here, like the typical adaptive immune response. So that would be easy to do, you could imagine. But how would you get all that rich different cell fates? And one possible way that could happen is like this, is that you use, again, the power of randomization. Just as you use randomization to generate those receptors, you could use that to allocate fates of cells. So here we've got the cell undergoing divide 5 times, stop and die. But as in each step, it spins a roulette wheel, and depending on what the individual number the cell gets, switch to IgG, become an antibody secreting cell, become a memory cell. By tuning, if you like, these probabilities here at each step, you can assign different proportions of cells into different fate as you go. And as a consequence of that kind of automaton, you can get the proliferation, cessation, and death. And along the way, you can generate different proportions of the appropriate populations. Okay. So using this sort of wind-up idea with the allocation of, or with appropriate use of randomization in appropriate places, you can achieve the same effect as all of that external control. Okay. Now, the important point about here is that this type of, if it was working this way, it's much easier to model. You can just capture those rules and set it in motion, and you can account for this interesting, rich population behavior without having to know everything that happens along the way. Okay. So, is there any evidence for this second idea? And I'm going to give you 3 examples. The first example is linked to the idea that as the cells are dividing, there's a probability, there's something changing as the cells are dividing that's affecting their fate. And we first found this when we started working with a dye that allows us to track cells through different generations. And we could follow cells as they're dividing. They're always heterogeneous. They take different times to divide. If we look at where they are in the division profile, in different experiments and at different times, it just makes a mess here. But if at the same time we use flow cytometry, we can stain the cells and ask what proportion of each cell of the population in each generation have changed into a different cell type, in this case expressing a different antibody type. All of these cells here are expressed again here on the same plot, but you can see now they make this incredible pattern where each generation, more of them become IgG2b. So these are the same cells. This is chaos, and this is a single pattern. As the cells are moving through division, doesn't matter how long they took to do it, the probability of them becoming this cell type is being influenced by the generation. Here's another example here of a different type of antibody. Many, many behavioural changes of lymphocytes that we see follow a division-linked pattern, just as would be expected in that automaton, that wind-up toy that we talked about. Here's another example, regulated division number. Here we've taken to filming cells and watching them in vitro. So these are now B lymphocytes stimulated to start dividing. So in effect, you're watching a little immune response in the test tube here. There's a little well here, and we're watching these cells over, sped up, let's watch over 3 days. So you can see the cells, imagining they're wandering around a lymph node. They start dividing here, dividing again, You can see, catch them, they divide there. This one's about to divide, I think. Maybe, if I can follow him. Come on. I'm letting this run because the behaviour gets into, there it goes, just divided. When we stimulate cells in this way, they seem to divide 3 or 4 or 5 times, then they stop. We knew that from watching the population, but by filming them and watching single cells, a student, Ed Hawkins here, followed individual cells, hundreds of individual cells over many days. There you see, eventually you can see they stop dividing and they die. The flaming, this is apoptosis, this is cell death, programmed cell death. They flame up like striking a match. So we've stimulated them, they've divided 3 or 4 times and they've stopped and died. And we can follow lots of features. We can look at the variation in times to divide. We can look at the inheritance of variation in division times. We know there's very little. But this plot shows some work that Ed did. He measured the size of cells through each generation. So here's a cell that divided twice. It grew, divided, and then it didn't grow. It just sat there and eventually died here. And this cell divided 3 times. Divide. Then it didn't grow anymore, and then it eventually died. This cell divided 4 times. So there's variation in the population as to how many times the cells would divide when you watch them. And you can see they divided, and eventually in their last generation, there's no impetus to grow here, and eventually they die. Now what's shocked us was when we looked at the family trees, entire family trees, they looked like this. Almost always, even though cells would take, in the population, they divide twice or 3 times or 4 times. There's quite a big spread in the number of times cells divided. But it turned out that all the progeny of a single founder cell here, this cell here, would give rise to progeny that all divided the same number of times before they stopped dividing and eventually died. Just as if they were wind-up toys. And something about this founder, so it wound up so much that it passed on to all its progeny enough petrol, if you like, to divide 4 times and then stop. There's other founders where it divided 3 times or twice. So that was strong evidence that these cells were somehow programmed here to run a certain number of divisions before they stopped. Okay. So the final example of how this might be working to allocate is allocation of fates. And this is a bit more complex. This idea that you're spinning a roulette wheel, if you like, and allocating fates in a certain proportion. And how would that work? Now, how that works, we've been looking at very intently for a few years. And we realised we would have to film cells and catch them in the act of doing different things and asking, well, how are the different fates being allocated? And can we find some pattern or rule to tell us how that's working? And so here's lymphocytes. We're stimulating them in vitro. We get them dividing. And as they're dividing, they're changing what they're becoming. And eventually they're dying. And we're going to follow, by filming these cells, 4 fates. Division, switching the antibody type they make, whether they become an antibody secreting cell, or death here. Okay. So this took quite a bit of manipulation to work out how to do this, but here's a single cell monitoring here. We just, if you like, the microscope is looking at hundreds of wells, and we're making a movie. We've just concentrated on a single well here. We go through the movies, find the wells where we watch a single cell, we watch it divide, and then we watch it to the next fate. Now, if it becomes an antibody secreting cell, these are special cells that go green. This is one of the immunology tricks. We can put reporters under a particular promoter. So BLIMP is a gene that's always turned on an antibody secreting cell. So when this is turned on, it also turns on green fluorescent protein, and we can detect that as cells going green here. Here's a technique where we use, if a cell switches to IgG, it turns blue, and we can detect that as well. So by watching these cells, we can watch them divide, we can watch them become an antibody secreting cell, we can watch them switch, and we can watch them die. And that's what we did. We looked at thousands of cells. When I say we, I didn't. Zoe did, and she's sitting over there if you want to ask her how she did it. But really, it took 3 or 4 months to do this, and probably a few years to set up the experiment in a way that we trusted the results. So this was hard work. Now, as a consequence, we got for the first time what the distribution times, we knew the times from division. This is time to divide. The time to the next division makes a nice strong distribution. Looks like a random process. Death times look like this. Becoming an antibody secreting cell, turning green, makes a distribution like this. We hadn't seen this before, and this is switching distribution here. And we sorted cells from different generation, and we could see that the time that these events took would change across generation, and the frequency which happened, frequency of switching, for example, as we saw before, is increasing with generation. So that all made sense. Why were we doing this? Okay. Well, it turned out the amazing thing, the point I want to get across here is when you follow single cells, every single cell does something different. They're all individuals, they're all unique. The time they take to do something, what they do along the way, will make its own particular pattern. Incredible heterogeneity of watching single cells. Nevertheless, every time the population, the same proportion will always divide, the same proportion will die, the same proportion will switch. So there's something going on where every individual is different, but the population behaves reproducibly. So when we looked at the individuals, we found interesting patterns. First one was that the times to divide, even though they're highly spread, you can see the time of two siblings were very similar. So there's a high correlation in division time between two siblings here. Pearson's correlation coefficient 0.94 here. This is time to die. Two siblings that die, high correlation here. So there's something shared between siblings. We also found interesting correlations in other fates. So here's time to divide here and time to die here, where two siblings were like this. There's some correlation, 0.6. Time to divide here, time to differentiate here, some correlation, 0.66 here. Interesting, really rich, interesting statistical features of what's going on in these individual cells. Now we had help from a really terrific mathematician, a probabilist, he calls himself, from National University in Ireland, the Hamilton Institute. This is Ken Duffy here. And the analysis we did together earlier this year proved very interesting about what's happening, and it was just published a few months ago in Science. So I'm going to try to give you a flavour of why this was judged sufficiently interesting for that. And it turned out that the hypothesis that we were testing goes something like this. Of those fates, we imagine the four fates were autonomous mechanisms inside cells. And that two of them, for example, are illustrated here. This is like an autonomous machinery for division and an autonomous machinery for differentiation. Both are working as if they're like a timer. But that timer, the time you choose for which this event should happen, is effectively the result of some random process. It's like the roulette wheel. We don't know what it is, but that random process is described by a probability distribution. So this individual has a time here that makes it fire here, but it drew that time from a probability distribution. So in the population, you'll get all sorts of mixtures of division times and differentiation times in the population. In this individual cell, though, the division time is set before the differentiation time, so this cell will divide before it differentiates. But in the population, we get all sorts of rich mixtures. So some cells, and of course the other point is that there's censorship. So even though the differentiation time that these cells are allocated might be out here, we will never know that because the cells will have divided before they reach that point. And once they divide, they start the process again. They choose a new time. So as a consequence, even though these might be the distributions that these autonomous modules are drawing from, what we actually see is shown here by the darkened circles here. We only see cells that differentiate before they divide. And as a consequence of that, we see correlations appear across cells. So here if we just plotted division time versus differentiation time in the population, we get this kind of pattern, and there's no correlation whatsoever here. But if we take these data here for the cells that differentiate then divide, because of the censoring that goes on here, now we get this pattern. Same data, but we get this pattern, and there's a relatively strong correlation between the data. That isn't there because there's any connection between the two processes. It's just a consequence of the competition that's going on for the two fates. And when we look for these features in the data that we had, where we had we imagined 4 autonomous modules, each with simple rules of censorship. Division restarts everything, death always wins, switching has no effect on the other fates. And if you become an antibody secreting cells, you no longer switch. These were the very simple rules. And it turned out that simple model could completely explain the data that we saw for those thousands of cells, where every cell was an individual, but it followed this very simple pattern and showed these interesting patterns of correlation across different concordant fates, concordant and non-concordant fates. And so that's why it turned out to be interesting enough for science to publish. It gives a really important clue as to where the heterogeneity is emerging from and how you control the allocation of complex proportions of different fates by just setting and tuning these probabilities of autonomous modules inside the cell. And as a consequence of that, as the system plays out, you generate any number, thousands of different individual fates, but the whole population is predictable and will do the same thing over and over. Okay. So this is coming to the end now. We have this idea that we have competing fates going on inside individual cells, and this can account for individual heterogeneity, extraordinary individual heterogeneity, but population consistency. We can build this into computer models and simulations now where we can accommodate the probabilities, how they change with generation. We can draw individual family trees where they're all different, but the population will follow consistent behaviour and generate cells of different types, all the different antibody types. So, just to summarise the reverse engineering, how to put it back together, I've shown you that immune cells show features of being programmed automata. That's not to say that they aren't influenced by external signals, they clearly are. But my view would be that they're doing things anyway, and that external signals can change the rate or the probability of how that's working. So it gives us a real clue as to how to dissect this very complex system now. And this, as I pointed out, is the much easier alternative for describing this complex system than this external control. And that the key fate machinery appears to be stochastic, and that the final result arises from competition, and that outcomes play out in population heterogeneity. Now, this can be used to measure genetic differences and drug effects and lead, hopefully, to predictive simulations and therapies, allow us to manipulate the immune system in some detail. And I just summarised there with the star there that this introduces a surprising role, I think, for randomness in cell fate and cell behaviour, in the same way that Burnett proposed for the generation of the receptor. So randomness, it turns out, is a key feature in many aspects of the lymphocyte life and in how the immune system works. Now, so let me just finish by saying this is really the biology of systems. And I think we have 2 great systems being studied in Parkville. This magnificent building's dedicated to the brain and the nervous system. But in the immune system, we have the Walter Eliza Hall Institute across the road. We have the new Peter Doherty Institute going up just across the street there. A real powerhouse, I think, for immunology between these 2 institutes and other institutes around Melbourne. And I just want to come back to my original story about clonal selection, that the head start that was provided by Burnett and transmitted to great scientists like Gus Nossel, Jacques Miller, Ian Mackay, Graham Mitchell, and many researchers in subsequent generations who invented and continue to invent amazing methods to reverse engineer this really complex, remarkable system, and hopefully in order to realise many health benefits in the future. So let me just thank, to finish, of course, Walter Eliza Hall Institute, where I work. Most of my funding comes from the NHMRC, and we get support from NICTA through supporting, in particular, John Markham, who helps us a lot in the imaging and image analysis and setting up the microscope. It's been invaluable, so thank you to NICTA for that. And Etsuko Uno and Drew Berry for that wonderful movie. Ed Hawkins, I think, who did I show data from? Ed set up the movies, Mark helped, Jay did all the counting, Cam helped set up the models, and Ken provided the probability experience and expertise. So I'll stop there, thank you.
I'd like to thank the sponsors that make this event possible. The Bio 21 cluster, the Melbourne Convention and Visitors Bureau, Phillips Ormond Fitzpatrick, IBM, the Melbourne School of Engineering, RMIT University, Monash University, the Bionics Institute, Bionic Vision Australia, the Centre for Neural Engineering at the University of Melbourne, NICTA, the Victorian Life Sciences Computation Initiative, St Vincent's Hospital and Melbourne Health. So please join with me in thanking our sponsors and in thanking our presenter, and we wish you and your team great success, Phil. Thanks, Phil.